Welcome to this month's PARC seminar. Just to announce the upcoming event. Next month, Dr. Raman from UCSD will give us a talk about harnessing health signals with large-scale mobile sensor data for modeling substance use disorder. Upcoming in a couple of days, in this coming Saturday, we will have the neuromodulation symposium. It starts relatively early, 7:30 and or 8 o'clock. It's at Mission Bay, the Neuroscience Building, right by the Rock Hall. And everyone is welcome to join us, either in person or either through Zoom. The Zoom site will be posted, the Zoom link will be posted, or if you register to this event, you'll be able to get the Zoom link for the symposium. And today, we're very glad to have one of us, Dr. Xiaobing Yu. He is associate professor of our anesthesia department. Xiaobing graduated medical school from China at the PUMC and get his postdoc training at Johns Hopkins and anesthesia residency at the Brigham and the pain fellowship training over here at the UCSF. He is currently NIH-funded researcher, a physician scientist, and he's today will present his very exciting work on the ACVR1 sensory neuron-specific signaling. With that, I'll leave to Xiaobing.
Thanks, Zhongwei, for the introduction. Thanks again for inviting me to participate at the PARC seminars. I'm going to share my screen. Are you able to see my screen? Okay, good, yeah. Thank you. So yeah, I changed the title a little bit. I think we're working on a manuscript. I think we are debating whether this is the information precisely presented. It's a great honor to speak to our audience, and also I want to highlight, I'm sitting in our brand new building and Bayfront building, and we have relocated from Mount Zion, which is dark and old, and to this brand new building. And when you have a chance, if you stop by, you should visit. It's just next to 1 block from the Chase Center. All right, so today I'm going to focus on this rare bone disease. It's not a rare, but disease. One of the genetic forms is rare, but heterotopic ossification is not uncommon, and often the pathology is that bone tissues and generate from the soft tissues after the injury, following injury, such as a blast injury on the battlefield, or people have the hip replacement, or someone suffer from TBI or spinal cord injury. And then by some unclear mechanism, and the bone will form from the muscle, injured muscle, and the connective tissues. And just imagine in the joint, if you have the bony ossified tissues wrap around the highly mobile joints, and it will cause a significant, the dysfunctional changes. And neither HO or its associated pain has effective treatments, and the underlying mechanism unclear. A subset of the HO is a hereditary subset HO, namely, and fibrodysplasia ossificans progressiva, also known as FOP, provided this opportunity to elicit a mechanism underlying to address those questions. And when I first called to service to, by my colleague and the current collaborator, Dr. Xiao, to see the patient with FOP, I had no clue, never heard about this. I had to ask, I was joking, I had to ask Dr. Google to tell me what FOP is. So now this is all the start. Inspired by the patient, we started looking into this, and eventually we become know more and more about this, and potentially can help us expand the scope of the beyond the rare bone diseases. And this is just an image to show you that normal bone, if you take X-ray just on the figure A, you can see it's just like us. However, in those patients, either when they are a kid, they bump into each other, and or they just in small injury can trigger the massive bone formation, and from the muscle, injured muscle, and the connective tissues. They often most commonly occur in the neck, spine, and scapular area. Just imagine when you have the bone form around that area, it's very difficult for them to become functional. And 97% of patients have been identified carry the single gain function point mutation, which is the R2XX mutation. So we started there. So commonly believed is that when we have a muscle injury, for example, this one, and if you have a muscle injury like incision and a burn injury, will trigger the mesenchymal subset of the progenitor cells in the muscle to proliferate, to differentiate and participate the repair. And the worst scenarios, and they will form the scar tissues. However, in patients with FOP, the whole stem cell change their fate. And because this is a subset of the mesenchymal stem cells express this gain of function mutation, so the BMP signaling is activated. I think I probably forgot to mention about this signaling is the type 1 ACVR1 is a type 1 BMP signal. And in this case, that the stem cell will change the fate, and working with other immune cells, they're going to become and bone formation. And just imagine, eventually, instead of repairing the muscle damage, and the bone will appear in the injured area. And we previously reported, we recruited cohort patients, and we did the quantitative sensory test and the golden clinical study to look at the pain. We found that the patient, all those FOP patients, their baseline heat hyposensitivity and the mechanical pain hyposensitivity in the absence of inflammatory flare, suggesting a neuropathic pain phenotype. And using the patient, derived fibroblast cells, we drive them into the iPSC cells, further differentiating them into nociceptive sensory neurons. And using CRISPR, the other technology, we demonstrated this mutation is sufficient and necessary for the hyperexcitability of those nociceptive sensory neurons firings, and the hallmark of some form of the neuropathic pain. However, still have the clinical question that has not been answered. For example, we still don't know whether in vivo action of this mutation in neuropathic pain is not been studied. And a lot of people believe in the field is that the pain is very common because 87% of the patients in the natural history study show they have a chronic pain. But often people believe it's likely secondary to the heterotopic ossification. And so the in vivo function has not been well studied. Another question is that we showed there is a changes in the nociceptor inputs and the morphology changes and functional changes, but unclear whether there is a nociceptor contribution to the trauma-induced HO. So in order to address this question, we utilize the preclinical animal model. So in this animal, and the human R2XX mutation was introduced. And we cross from PDGF alpha, which is expressed in the mesenchymal stem cells. And when we introduce an injury by a steel ball, and on the quadriceps muscle, the thigh muscles, and in this case is demonstrated, in 2 to 3 weeks, the mice will inevitably develop ossified tissues around the femur bone. And you can see by looking at their changes and looking at the gait changes, they are significant different. And this is just a video show how do we do this. This is a assembly of this ball drop device. And this is Liam to show demonstrate. So the pins directly contact the anesthetized muscle quadriceps and to introduce the closed injury. All right. When we look at those tissues, we always wonder is because most of the people just is a bone, and we just using a micro CT in the CT and the imaging like H&E stainings. And we did it differently, is that we just decalcify the tissues and then we stain them. I was surprised. We found there is a significant nociceptor sprawling in those ossified tissues. And you can see is a magenta color labeled by both PGP 9.5 is a pan-neuronal marker. We further confirm with CGRP and nociceptor markers. And then the question is that, is this unique to the animal model, or we find the same thing in the patient population? And it was grateful to have patients of FOP patients when they passed away, they signed the informed consent and donated their tissue to us. So we're able to access their tissues, and one of them is they have a clear chest wall heterotopic ossification. When we decalcify the tissues, and we're able to see there is a also significant nerve sprouting within the ossified tissues. So then this is interesting finding, and to suggest there might be a contribution of this potential nerves and the nociceptors to the HO. In order to study that, we trying to selectively silencing these nociceptors in the muscle before the injury. So what model we use is that we always just briefly just talking about how the nociceptors behave. And nociceptors, mainly the small unmyelinated fibers, and spread all over our body, including skin and the area. And the multiple, the important mechanosensory, the receptors expressed. For example, TRPV1 is expressed, TRPA1 is also expressed, and the other receptors can sense the acid stimulation, and the piezo2 has been known to respond to the mechanical stimulation should the amylase require 8. And many of the studies actually found at UCSF. And most of you already know this TRPV1 finding at piezo2 got a Nobel Prize, and David Julius is one of those the Nobel Prize laureates. And when the nerve injury, tissue injury occur, and the DRG neurons is where the somatosensory neuron cell body located, can become haywire, and they can send the signal to the spinal cord and further send the signal to the brainstem, to the cortical regions, so we can feel the pain. And interestingly, this, the communication can be bidirectional. In addition to sending the pain signal to the central nervous system, and the signal and also can send by sensory neurons to the peripheral. For example, in this case is that some of the specific nociceptors can release neuropeptides or other neurotransmitters. And in this case, they can attract the immune cells to the injury site, and which are causing the neurogenic inflammation and heat, red, and the pain and the swelling. And in clinically, you can order in the preclinical model, there's several ways to block the release. One way is that you ablate the nociceptors. Mainly we use the capsaicin or RTX is a TRPV1 superagonist. They're causing the overflow of the calcium channel, flow, and then causing the ablation of the nerve. Another way is a Botox. And the clinical ways of Botox injection to treat migraine headache, or some cerebral palsy patient, we're using the Botox to relax the spastic muscles. The part of the mechanism is that the Botox able to target the SNAP25 complex and silence the nociceptor release neurotransmitters. And in this case is that we know when you give a Botox in the intramuscularly, we expect Botox to silence the neuropeptides release. But at the same time, it is expected and acetylcholine will be blocked as well. To confirm or to optimize the dosing, we want to also, we first try the dose and all the mice died. And to treating optimize the dosing, and we're using the different approach. We just we follow the mice, and particularly looking at the gait changes induced by the acetylcholine blockade. And we're using this black box developed from Boston, and we have collaborators, and they're very experienced to do the analysis. So in those black box, mice allowed to freely moving, And there's a light source into the smart glasses. One is just a transluminance LED light to capture the posture and the position. And the other one's FTR, the infrared camera, capture the pressure on this illuminance to indirectly to reflect the pressure of the pulse on the glasses. So you can see from here, in brief, is that we look at the animal behavior and what the posture changes. And you can see from C and D and E. And after the Botox injection, and the mice definitely travel less compared to the vehicle sitting group. And also we notice is that after the Botox injection, after acetylcholine release is blocked, and the mice start to shift their habit, they put more pressure on the contralateral side. And you can see from the D as well. So using this approach, we're able to come out a very, find the dosing, which is a safe and effective to treat the mice. Then we inject the same dose. This is all the white type mice. And then we inject, using the same protocol, we inject an FOP mice. And you can see from the left panel, if you give them the saline injections after 2 to 3 weeks, after the ball drop, mice will develop the massive ossification around from the quadriceps muscle, wrap around the femur bone and the tibial bone. Interestingly, when you give a Botox injection, we inject 2 doses, and 2 hours before the ball drop and 24 hours after the ball drop, we found there is a significant reduction of the ossification in those animals. If you look carefully, it looks like even some of the animals develop the ossification, but the texture and those ossification quite different from the solid status of the vehicle-treated mice. So to further support the functional connection between HO and nociceptors, we utilize the TRPV1 superagonist, which is RTX. RTX is 100-fold more potent than capsaicin, and there was a lot of studies show it's useful. So we have to do the systemic injection. Interesting is that we realize is that when we inject those mice, a lot of those female mice died during the injection. So we have to utilize conduct a study in the male mice. And we found out if you're given about RTX 5 daily IP dosings and 3 weeks after the ball drop, we found the the TRPV1 afferent ablation prevent HO from formation. And this is just a quantitation. And in the Botox, we look at both male and female mice. And RTX, all this data presented here is the males. So we conclude nociceptors are necessary for the development of trauma-induced HO. And we encourage by the result, because then we question is that we want to address if we introduce the mutation, because FOP patient, all the cell types, including bone stem cells and neural cells, all express this gain function mutations. Then the question is that if we just introduce mutation into the sensory neurons, can we induce HO formation? So what we utilize is that we introduce this human RTX mutation into the Edwin-Cree ER mice. Edwin-Cree, many of them know, is a sensory neuron marker and also mark the synthetic neurons as well. So we allow the RTX activated in all the sensory neurons. And then we repeat this, ball drop injuries. We look at them carefully. And to our surprise, we did not see any heterotopic bone formation in those animals. We conclude the mutation in the sensory neurons alone is not sufficient to induce HO. So you need nociceptors are necessary, but you need a mesenchymal stem cells to participate the pathology. All right. Another thing from the literature we do know is that macrophage are the main immune cells, and in the following the soft tissue injuries. In FOP particular, there are a lot of studies published from my collaborator, Dr. Shell's lab, is showing that, and the increasing level of the systemic inflammatory cytokines release is a hallmark of this FOP flare-up. And when they isolate the patient, immune cells, meaning the macrophage, they are they become primed pro-inflammatory. When they isolated using the patient iPSC cells to drive the macrophage, they found that many of them is M1 type. And UPenn group, actually, they trying to use a collagenate to deplete the macrophage in another model of the FOP animals. And they find out if you collagenate to deplete the peripheral circulating macrophage, and they observe the reduced HO formation, you can see from here. So we're interested in this. We wonder is that whether the muscle nociceptors potentially can regulate macrophages. So it is very hard to do this experiment in the FOP mice because you're dealing with the bone structures. So we utilized the wild-type mice. We repeated the same injection. We injected the Botox before, after the injury, and we collected tissue 3 days after the ball drop injury. And in the contralateral uninjured muscles, we do not see the massive significant macrophage infiltration. In contrast, on the injured side, we see the massive infiltration of the macrophage, which is labeled with IBL1 and F480, the macrophage markers. And however, we notice there's significant reduction of macrophage infiltration in the Botox treatment group. So suggested the muscle innervating nociceptors are involved in macrophage infiltration. And we further look at the FOP mice. And before we collect the mice for a micro CT analysis, we also save the blood for multiplex cytokine array. And we find out there's a lot of those, we particularly focus on the cytokines, the pro-inflammatory cytokines and the macrophage recruiting chemokines. Then you can see from this figure D. And notably, CCL2, the primary responsible macrophage recruitment, was significantly reduced. And another markers, we find out is IL-1 beta. And IL-1 beta has been commonly, detected, increased levels are detected in FOP mice during the flare-up. And the clinical case report did show is that if you're using the anti-IL-1 therapy, using the receptor antibody or directly into the IL-1 beta, we can significantly reduce, the FOP flare. And we found that RTX led to reduction of the IL-1 beta level systemically. And also when we look at the DRG, we also find out only in the FOP mice, there is increased macrophage numbers by detail and highlighting with CX3CR1 macrophage markers by RT-PCR show there is increasing expression of the CX3CR1 only in the ipsilateral, which is injured side FOP mice, but not in the wild-type mice. So we conclude that RTX of Botox involved, the reduction of the HO is likely associated with reduced macrophage infiltration. So I just to demonstrate here is that we recorded a significant nociceptor sprouting within the trauma-induced HO in FOP preclinical model, as well as in a patient FOP. And we also demonstrate that nociceptors are necessary for trauma-induced HO in the FOP preclinical models. And as I mentioned earlier, is that we're the pain lab. We're always interested about the pain. And I mentioned the challenge we did show there is a lot of the evidence we published from human data and suggest there might be independent neuropathic pain phenotype. But we just don't know is this a secondary or this is a primary. So to address that, we utilize the edwin mice to selectively induce the mutation in the sensory neurons and look at the pain phenotypes. And in this case is that we're using the series of the pain studies and look at the animals in a naive state, at a stable state in the absence of the nerve injury or tissue injury. And we find out that because it's tamoxifen-induced model, And we look at the baseline without tamoxifen induction, and we find that there's no difference between the two groups, the Edwin-Cree control group and Edwin-Cree ACVR1 mutant groups. However, when we inject with tamoxifen, induce recombination, and we find out, and the mice who carry the ACVR1 R2X mutation and can develop the hyposensitivity, even you stimulate them with the softest, lower long-free filaments on the hind paw. And we observe the both pattern in both males and females. When we look at the heat pain threshold, when we put on the heat plate, and the mice only can tolerate for a certain period of time before they try the licking hind paw and jump out of the cage. And then we find out after the tamoxifen induction and ACVR1 R2XH mice, had a clear lower heat pain withdrawal latency. So our data suggests there is a hyposensitivity, and in the stable status without the injury, without the tissue or nerve injury, suggesting there is at a naive status, they have a baseline neuropathic pain phenotype. And then we're interested in what could happen in those mice. So we using the RNA sequencing, the bulk RNA sequence, to isolate the DRG from the two groups. And you can see is that from the figure A, there was remarkable transcriptomic changes in both, when we compare both groups. And notably, we find there are several macrophage markers was significantly induced by selective expression of the R2XH mutation. And we further confirm using the RT-PCR and to clear the CX3CR1 expression is induced in this group. And we also look at the ATF3, a marker, it's a stress marker, and most people using to define the tissue injury. We find there's no change, suggesting the increased expression of CX3CR1 in the DRG is not induced by injury. And when we're looking at the protein level, when we stain IBA1, another macrophage marker, and we can see the significant increased expression in the DRG. And if we look at the morphology, they're quite different. And we quantitate them and confirm there's the changes. And also, when we run the FACS analysis, we're trying to see the quantitatively given the cell number difference. Because sometimes the cell number may not change, but it's just expression level can go up. And we further confirm is also that in the R2XH led to increased numbers of the macrophages in the DRG. And previously, we also look at the spinal cord, and you can clearly see is that the R2XH expression of sensory neurons did not lead to any microglial changes in the spinal cord. And previously we showed there's a nerve injury can induce the DRG resident macrophage to proliferate, contribute to the pain pattern. So we're using similar strategy, and we label those cells with both CX3CR1 to mark themselves as macrophages, and also we do intracellular staining with Ki67, which is proliferation markers. And we noted there's the percentage of the cells did not differ between two groups. So we conclude the DRG macrophage increase in this ACVR and R2XH mice and does not result from local proliferation of the resident tissue, the macrophages. Rather, we hypothesize they come from infiltration. And the recent study from other groups, from Japanese group, they did show is that in the absence of the tissue or nerve injury, and the peripheral macrophages, mainly in the skin, the other areas from circulation, can contribute to the acute nociceptive changes. So we use this strategies that if they're coming from peripheral, if we deplete them with collagenate, and then we see what happened on the mechanical threshold. So collagenate systemic injection have shown is that they only target the peripheral circulating macrophages, but spare the DRG macrophage. So using this strategy, we successfully limit the mature circulating macrophages. Then we examine those mice, and we find out the depletion of circulating macrophage led to increased mechanical threshold, particularly in the ACVR and R2XH mice. And subsequently, we just wonders what could contribute, what could influence the infiltration of the circulating macrophage to the DRG. And the recent the study from Boston group, and they shared is that macrophages are the most important immune cells interact with sensory neurons, and in particular the peptinergic neurons. So we hypothesize that if we introduce these genes into the non-peptinergic neurons, which are known to release less neuropeptides, and likely we're not going to see this, the increase of the macrophages. So we did an experiment, and we specifically introduced R2XH mutation into the MRGD, which is peptinergic neuron markers mice, and we did not observe increased macrophages. So we conclude, and very likely the macrophage expansion in the Edwin-Cree ACVR2XH is likely rely on peptinergic population, a subset of peptinergic neuron populations. And a lot of the paper did show that CGRP actually can directly regulate the macrophage migration by binding to its cell surface markers. So then what we did, and finally we did, is that we just isolated those sensory neurons and do the in vitro culture. And we find out that only the R2XH animals have increased capsaicin-induced CGRP release using the ELISA. Consistently, our data suggest this sensory neuronal R2XH expression can induce peripheral macrophage to the DRG. And in the previous published work, we did show is that ACVR1 actually express all sizes of the sensory neurons. And interestingly, after the nerve or tissue injury, ACVR1 induction only observed in the ATF3 positive neurons, which means they have a nerve injury. So then we ask is that because the FOP is a very rare bone diseases, we wonder is that can we see the potential in the more common neuropathic pain conditions? So we use the spare nerve injury model commonly used in the lab, and we utilize this ACVR1, the kinase inhibitor called LDN 19066, and we do intrathecal injections to only target the spinal cord and the DRG cells. So we don't expect it will impact the microglial cells or the other, the ACVR1 population, mainly the DRG. And we find, using the blocking this ACVR1 signaling, after the nerve injury, can prevent the neuropathic pain formation. And in the chronic pain conditions, after 3 or 4 weeks after nerve injury, and we see the partial reversal of the mechanical hyposensitivity. And rather solely depend on this is more reflexive withdrawal responses. And people in the field arguing about the translational values. So then we using the black box to assess the spontaneous pain behavior evoked by walking. And we use different drugs like morphine, IP injection, gabapentin injections, and LDN drugs. And we did see their analgesic efficacy is similar compared to morphine and gabapentin treatment. So our data suggests that targeting the neuronal ACVR1 signal using the LK2 or ACVR1 inhibitor potentially can be used to treat more common neuropathic pain conditions. As I said, is that previously we did a show is that nociceptors are quite important and necessary involved in the HO mutation, HO formation in FOP animals. And we also show is that if you just introduce selectively induce R2XH mutation in the primary sensory neurons, by itself can provoke neuropathic phenotype in vivo, which is independent of the HO or tissue injury. And the last batch of the data further support pharmacological inhibition ACV1 LK2 may reduce the neuropathic in the Y-type mice. And encouragingly, there is a current highly selective ACVR1 or LK2 inhibitors already in clinical trial in the FOP patient to particularly focus on the HO formation. I was able to participate in the study, but my focus mainly on the pain. So we are testing those new highly selective drugs in the other neuropathic models, such as trigeminal neuralgia models, and to figure out whether they have a more broad impact. And so we conclude. So our findings uncover a primary sensory contribution to the induction HO and to a neuropathic phenotype in FOP. Together, we review the potential therapeutic target to address these two unmet clinical challenges. And so hopefully we will have more data to show you in the future. So this is just my lab members and my collaborators. And many of them know that Dr. Basbon is my mentor, and we continue working closely. This is during a Halloween, a party, you can see them then. And Dr. Shao is an expert in the endocrine and the bone diseases. So, the study is supported by the including intramural, foundational, and also the NIH federal funding support. So to end my talk, I will use this slide. And for many of us doing the translational research, we always struggled. How do we improve the translational success? So this is a photo I took from the Vancouver area when I visited. And just to tell you this, the kind of a challenging or dilemma we're facing. And thank you. So I'm open for questions.
Right. Questions? Anyone? All right, Xiaobing, now I'll initiate. So what do you think the potential mechanism of the ACVR1 in by itself to reduce pain?
Okay, so we have some of the evidence from other things I did not elaborate. And I think the stimulating the macrophage infiltration potentially is a role. And also there is other potential target. We find one of those receptor involved in the gabapentin binding is also impacted by this mutation, and still a lot of answers. And interestingly is that we also find some subtype specific genes also impacted. Interestingly, not all the way induced, they are repressed by this mutation. So you can imagine, because when this is the BMP signaling is massively induced, so it's not going to only impact one subset of the neurons, might impact the broad spectrum of the changes. So we're currently working on that, trying to better understand the mechanism, but it doesn't look like it's just one single mechanism. As I showed, there is probably multiple mechanisms involved.
Okay. Peter has a question. Wouldn't the nociceptor sprouting within the trauma-induced atrial imply the presence of central sensitization in injury-induced heterotopic ossification?
Okay, great question. So it's always intriguing to see neurogenic heterotopic ossification, mainly associated with the spinal cord injury TBI. And the mechanism really unclear because most of the patients do not carry this single point mutation. We know that. Animal models also intriguing. So spinal cord injury alone is not going to induce HO. You have to do the muscle cardiotoxin injections, which is manifested with a massive macrophage infiltration. And but in those studies, I don't think anyone particularly looking into the heterotopic, so what's going on there. I think the part of the reason is because bone tissue and the decalcify the bone tissue is always challenging. Like in the human sample, for example, it took me a week to decalcify them. So it might be just limit our opportunity. But now I think we have more incentive to look into it. And the other thing is that what other components contributing, we don't know. I think it's still a lot of unanswered questions. And some people argue is that the central neurogenic one may also cause leakage in the blood-brain barrier, which may also promote the more peripheral effect. I don't think it is a significant contribution from central alone, but I feel the mainly is probably still the peripheral central impact.
Okay. Any other questions?
Yeah, I have a question about opioid receptors. On mu-opioid receptors in this inflammatory situation, there are mu receptors, let's say, on nociceptors, and they get activated basally by inflammatory processes, and that initially diminishes some of the pain, but eventually I think it turns into an inflammatory process itself. So have you looked at opioid receptor activity there?
No, I think that it's just too challenging to study, but I think it's a great suggestion. And I think I usually try to stay away from the opioid receptor, and I think it's just too complicated. But we have a lot of the better groups, they know how to study that.
Okay.
Why you have a question?
Yeah, I'm not familiar with this disease. I just want to clarify if I understand everything. So the ACVR1 mutation is a gain of function or loss of function mutation?
It's a gain of function.
It's a gain, that's why I see.
Yeah,
so
it's a
it
gain of
seems
function.
that the mutation has to be in the mesenchymal cells to induce the bone, the osteo phenotype. But then for the pain, the mutation in the nociceptor receptor neurons will be enough to have the pain, right?
That's right. I think your question, your interpretation is right. So in the past, people are always thinking about pain is usually secondary because the first pathology and the bone changes is most visible in this patient. That's why they prompted them to see most of the kids, they bump to each other, then there's a lump develop in the back, and prompted them to see a pediatrician. They are so scared that they think, is this leukemia? So usually they did the biopsy. And of course, the pathology results came back as negative, but they realized that the lump is getting bigger and bigger. Then that was the moment they think it is not regular, so it is a genetic disease. And so often is that when they have a flare-up, they always have this kind of inflammation changes. So people often believe it is secondary. But we showed the evidence in humans, they have a baseline difference. And now we presented the evidence to show is that without an injury, without HO, if you just introduce a mutation sensory neuron, is sufficient to induce a phenotype. And going back to the HO, and it's a gain of function because it's a BMP. Signaling is activated. And most of traditional people believe is a mesenchymal style fibroadipocal progenitor cells are the primary cells to respond to HO formation in this condition. And I don't think I can say, believe it might be true in the other non-FOP HO conditions, because there is a bone injury, as we said, a spinal cord, the central focus, derived, injury induced. But the data we showed here is that without nociceptors, they are not, the HO is not going to form. It is not sufficient, but necessary. I think that's our key findings from this preclinical. But whether it's going to be true in humans, we don't know. I think, but we're only able to draw the conclusion from preclinical model.
Yeah, that's very, very interesting result and data, and you did a good presentation. I have one more questions. So, whether those patients have a basal level, low level of threshold to pain, or do they actually already have some inflammatory cells in their dorsal ganglia, the high level than normal?
I don't think I have a definitive answer for that. The only thing we showed is that in the adult population, cohort patient we published and 2 years ago, and we compared their family members, which has a similar genetic background. We show they are more sensitive to the mechanical pain stimulation. We're using look at their threshold, and also their heat pain threshold is lower compared to their peers. After we adjust the sex, age, and to compare together. But interestingly, the cold pain threshold did not differ. And which we also saw in the animal model, we did observe the similar patterns. Then going back is that whether there is infiltration in the animals, I think now we start to have this donated tissues from the deceased patient. We may be able to look into again, but the problem is, what is my best control? It's always tricky.
Yeah. Have you tried to check the sensitivity of the patients with gene mutation, but did not have the ossification change yet? So
that's a great question. I think almost all the patients we encounter, all of them have ossification. So yeah, we haven't. I think I was told that there is some mild bone phenotype patient, and we're very interested to meet them because it might just have a totally different mechanism involved. But so far, all the patients in our study, they all have a significant disability because of a heterotopic ossification.
thank you. It's a very nice talk.
Thank you.
Any other questions? Alan, are you saying something? You're muted. You're muted, Alan. You're muted.
I know. Sure. Would you ever use RTX in a human?
Alan, I think I will let you to elaborate on this. I know you have probably I would not, but I know you have a different opinion on it.
I bring it up because Mike Iadarola is definitely, they're running clinical trials with RTX in cancer pain patients and in trigeminal neuralgia. They're going to propose to do it in trigeminal neuralgia patients, to go after dorsal root ganglion. Now, I suppose the problem in the patients is that you might solve one problem, but then if they injure themselves, they're going to get it somewhere else. So there's nothing really that you can do unless you can somehow do it pharmacologically. But then you need long-term treatment. Is there any a priori way that you could treat a systemic condition like this? What would be your choice?
So I still hesitant to adopt the RTX idea in human, but I would think differently. I think it might be productive instead of targeting this condition systemically. I'll focus on the focally. For example, most of the profound pathology in this patient is a FOP flare-up. I think exclusively end up with more bone formation. So given the data we see from the Botox, and I was wondering, I talked to the patient, actually quite a few of them, they're quite interested in this potential. And we can inject a very low dose. We currently already those doses used in the clinical for migraine. And when they first spot flare, we inject to the site and see whether we can prevent or limit the scale or the further development of flare. And the end point is that are we going to see more ossification formed? I think that's easy to determine, and I think the risk will be much lower. We don't need to worry about the thermal regulation, dysregulation from RTX systemic use. So I think the risk is higher, and also it's easier to get RAB approval because we're using, because the worst scenario, even it didn't work, because most of the time flare-up is irreversible.
And so what I'm still puzzled about is, where does the CGRP come in? You think that's just in the dorsal ganglion or in the peripheral terminal release, part of neurogenic inflammation? And if that's true, is there any value to carbamazepine or some of the CGRP antagonists?
I think it's great. I think I probably would say both, but giving that a massive nociceptor sprouting, I suspect is at least at the muscle injury site. It has to come from a peripheral release.
Okay,
And
so do you think, is it a vasodilation component? That seems unlikely. Or is it possible the vasodilatation is allowing the chemistry of the muscle to change, all the cytokines, chemokines coming in? Would Tegretol be of any value?
I don't know. I think most of people tried other neuropathic medication. It's become increasing standard. Actually, we kind of like give it a talk to the community, and we increasing using it. And also there is
you mean gabapentin?
Gabapentinoids, yeah.
What is that? What does gabapentin do? It'll treat the pain.
It's mainly the treat pain.
Does it prevent the ossification?
I don't think there is any evidence.
Okay, I wouldn't think so. I'd be surprised.
Yeah.
Okay.
Carbamazepine, it's, you're targeting the sodium channels. I think the problem is that I think the utility might be limited because in this population, they already suffer a lot of things. So the side effects associated with carbamazepine might be a hard sell.
Mm-hmm.
So in this case, is that I still going back to the Botox. I still feel there is a Botox utility in this, because Botox did show is that it can block CGRP release very effectively. That's most likely the true mechanism.
No, fair enough. But what about, there are you got the Boehringer CGRP antagonists. CGRP the antibodies might be problematic. They might not get in, but there are CGRP antagonists against the receptor.
Yeah. So currently, we're trying to confirm. We're currently ongoing experiment to treat the mice systemic with anti-CGRP, the CGRP receptor antibody. We're still waiting for the result. Intriguingly, and it looks like we prolong the survival of the animal. We're trying to confirm that's indeed a fact.
I would think that's a pretty easy observation.
No, well, the problem is that it's still they're sick mice.
No, I'm kidding.
So I don't want to just jump in and conclude, but I think I will probably next year, probably in this next 3 months, I may have better answer. So I think if you ask me to bet, I think the CGRP is a very reasonable target. But at the same time, I think we have seen, especially in a lot of the recent, we see a flood of those papers talking about amazing impact of CGRP involved in the sepsis, involved in the infection, involved in endometriosis and migraine headache, all the things. I just don't know whether we can translate into human. I don't know. That's the last slide I show is that it's always the kind of struggle we're facing. But I think it is important to know. So that's why we'll continue doing the experiment, because we're trying to accumulate enough sample size to conclude. But that's ongoing experiment.
Thank you.
You're welcome.
Xiaobing, just a follow up with the Alan's point. So in your experiments, you do the RTS injection systemically, right? IP.
Yes.
Right. So why you're not doing the local injection in the muscle, just like you do the Botox injection, especially the trouble with the female mice?
Okay, so I think we already have the Botox data, right? So it's always, your concern is that, are we going to completely ablate?
No, I'm not saying you should not do. What I'm saying is, why not do
Why not?
the RTX locally rather than systemically, right? I'm just trying to follow up the Alan's question.
Okay.
Right. So
we all know is that I think it involved another round of the painful RTX dosing titration, because we do know that even I injected, when we first do the Botox, I thought it was a very low dose, and but inject all the mice died. I think part of the reason is because, remember, this model is a ball drop. When you're ball drop and you are causing the muscle injury, which also causing the vascular leaking. So if you give the dose, even we think it is a we locally inject, there is a high chance, I'm pretty positive, they will be absorbed systemically. So we end up the same situation. And I just cannot easily kind of rule out that possibility. If we see the effect is still systemic, I don't think I can confidently say it's a local impact. But Botox, and at this moment, we're pretty confident it's mainly the low, because the mice did not die.
any other questions? Suggestions? All right. So I guess we'll end up here. Thank you everyone for coming, and hopefully see you guys Saturday and next month.